Hello, and welcome to the Human Microbiome Part 2. In this lecture, I'd like to tackle a few myths about the human microbiome. Also, we will cover a spectrum of various external and genetic factors that can influence what resides in or on our bodies. Also, I'd like to go a little bit deeper into the connection with HLA or immune mediating genetics. It's important that we recognize that microbes that exist in and on our bodies are interacting with us constantly. And so I also included a couple of slides where we can unpack some of the implications that are the constituents of our diet, for instance, can play in establishing certain bacteria that are more likely to reside in our gut. So, without further ado, let's get started. So we can get through all of these slides together. A few myths about the microbiome. Diversity does not always equal health. Although diversity is often associated with a healthy microbiome, some conditions may be associated with increased diversity, which may not necessarily be a good thing. It is the type of bacteria that matter more than just their number. And having more bacteria isn't always necessarily better. Balance and diversity are the key. And an overgrowth of certain bacteria can lead to health issues. Probiotics can be beneficial, but not all probiotics are equal, and effects can certainly vary based on the individual. Some may not have very significant effects on gut health or on the other intended consequences. Or it could be the case that taking excessive amounts is what's necessary and doesn't always lead to better outcomes. Our lab actually does a bit of research on this, and we believe that it is the efficacy of probiotics is in part based on our genetics, which can thereby influence what can and cannot colonize very easily in a person's gut. And while antibiotics disrupt the balance of the microbiome, the effects are not always permanent. The microbiome can recover over time. However, repeated or unnecessary antibiotic treatments can have much more lasting impacts. This is something that our lab also studies rather extensively in human pediatric cohorts, especially where we found that repeated antibiotic use appears to have connections to various autoimmune and neurological disorders. While a healthy diet is important, individual responses to diets can vary. There is a lot of literature, epidemiological literature, some clinical trials on various diet interventions, and they don't always work the same way in different people. Part of this could be due to genetics. Part of this is due to other mitigating factors like lifestyle and socioeconomics and just a whole wealth of variables. However, in a sense, it's also impacted by the constituents of our gut. So it's a pretty dynamic process as we change what we eat, how that impacts what bacteria are able to colonize because they're expecting or they require certain constituents in order to survive. What's considered very healthy or optimal for one person may not be suitable for another person's microbiome also. So this is something important to recognize. Not all bacteria in the gut are harmful pathogens, of course. There are commensal microbes which play very vital roles in digestion, nutrient absorption, and immune function. And again, it's that balance that is so important. And you another myth is that we can quickly change your microbiome. That's not always the case. Some significant changes take time and consistent effort. And quick changes, like we had just talked about diet, very quick changes may not lead to lasting results. This consistency is often what is required. The microbiome, human microbiome, is not limited to the gut. While it's more extensively studied, other parts of our body also have microbiomes like the skin, the mouth, the elbow, even the lungs. And these can certainly influence health too. Our lab has done, for instance, some work on the saliva microbiome. Of course, we're several weeks into the course by now. You should recognize that the microbiome refers to a diverse community of microorganisms that reside in a certain niche. And in this case, it's on and within the human body. These microbes influence how we harvest energy from our diet. They impact a host of things, not just weight regulation, adipose tissue development, but even behavior. They play a role in nutrient absorption, immune system modulation, and protection against pathogens. Interactions between host and the microbiome are extensive, playing crucial roles in early life and immune system development, contributing to long-term immune health. Also, dysbiosis can impact the gut-brain axis. This is the mechanism by which bacteria, or microbiota rather, can communicate with the central nervous system. And dysbiosis is associated with a variety of diseases and different health conditions, which you should now appreciate after having gone through that review article that we posted on Canvas. And this is just one example of an article that has summarized various conditions in which the microbiome is implicated. Metabolism, of course, the gut microbiome especially can modulate drug metabolism, affecting, for instance, the efficacy and side effects of different medications. And microbial metabolites, including precursors to neurotransmitters and even some vitamins, can impact brain function, immune responses, metabolism, a whole variety of different important physiological processes. Gut bacteria metabolize direct components of the diet, for instance, proteins, lipids, fats, carbohydrates, dietary fiber, plant polyphenols, resistant starches, simple sugars, polyunsaturated fatty acids, choline and lecithin, inulin and oligofructose. And each of these, I've listed here some primary diet components that you may be familiar with, or you may have as staples in your own diet. And these food groups, these specific constituents can have been shown to be metabolized by certain bacteria. So Faecalibacterium prausnitzii, for instance, requires a lot of fiber. Or it's a bacterium that can thrive off of that. Bifidobacterium will be able to metabolize different proteins. So what I think is interesting too is when we get into simple sugars, firmicutes and Bacteroides, Escherichia coli can metabolize these sugars. So it's important for you to, as you're navigating the literature and finding certain bacterial associations, think about who are these bacteria? What impact might they have on inflammation or on metabolism? What do they require to thrive? What sorts of conditions or what precursors? And what are their effects? So what can they then produce? Right. So I think that that's really important when we're considering the dynamic interplay of host microbiome interactions. And I have these two slides here just to further probe you in that way. So you can think about that when you're seeing different taxa thrown around in the literature, especially when they're important in an association. Factors influencing the microbiome are very vast, and I just included a few on this slide, such as medications and antibiotic use, diet, as we've talked about, lifestyle, environment, environmental exposures, toxins, pollution, geographic location obviously is an important confounder, as we see it's an issue that comes up when something might be associations may not be found if there's a lot of geographic diversity until the geography is accounted for in some way. So that's something that has been really important in studies like TEDdy, for instance, determinants of type 1 diabetes in the young, which has various locations for sampling children across time. And that factor of geographic location is really important. Of course, ancestry can also be important. And then there are factors like stress and psychological factors, things that can potentially impact inflammation and the immune systems. And often these can go hand in hand with gastrointestinal problems. Now, I want to acknowledge something very important in the context of the human microbiome is autoimmune disease, inflammation, and the HLA, or the human leukocyte antigen. Autoimmunity, for those who are unfamiliar, is linked to a lot of different mechanisms. These include mutations, molecular mimicry, where for instance, in the case of multiple sclerosis, where the immune system attacks myelin, and that's possibly triggered by molecular mimicry with viral antigens. So there's some really interesting literature that has emerged linking multiple sclerosis potentially with Epstein-Barr virus. If you haven't looked into that in the literature, it's quite intriguing. There was a group that harnessed data from the Veterans Affairs, very big study, and they found that people in this cohort with MS had all had Epstein-Barr virus at some point in their life. And so this has triggered a lot of very interesting, rich literature in this area. And as you can imagine, this can be important thinking about vaccinations and different targets to prevent autoimmune disease, right? So this is molecular mimicry. Epigenetic changes, especially if they lead to inflammation in the body. This could be the case in rheumatoid arthritis. And then epigenetic factors, for instance, exposure to PFAS, potentially in the water or other contamination in females especially, is highly linked to the progression, or rather the development, of celiac disease, whereby gluten is a trigger for the immune system to begin attacking the intestines of affected individuals. The cure is to avoid gluten or gliadin, the protein. But this is a case where epigenetics appears to be very important. For instance, sex dependent, specifically in females, and exposure to a chemical like PFAS can actually trigger the onset of the disease. Potentially, this is just another area where environmental exposures and epigenetics might be part of the equation. This is a nice figure to remind us of molecular mimicry. It's a similarity between self-antigens and microbial antigens, for instance, and that can thereby trigger an immune response. Genetics and environment play a role, but especially biological sex in various autoimmune conditions. We see this is a nice figure demonstrating in specific autoimmune conditions. There's a very big sex imbalance, whereas in others, it's sort of a toss-up, for instance, with Crohn's disease or with type 1 diabetes. But Sjogren's syndrome and Hashimoto's are much more prevalent in women than in men. Autoimmune disease incidence is increasing over time. This is any autoimmune disease, and this is broken down by socioeconomic status. So irrespective of that, even if we look within each of these separately, we see a trend whereby autoimmune incidence is increasing. We see this in celiac disease and in Graves' disease, and in rheumatoid arthritis, for instance. So MHC molecules, major histocompatibility complex. This is really important in the context of the immune system. I want to touch on this, especially since our lab is really interested in the role that HLA has in bacterial colonization. MHC molecules are surface cell proteins that play a central role in presenting antigens to T cells. This is crucial for the immune system to properly recognize both self and non-self antigens appropriately, or the material that's brought to our immune system for us to then deal with things in the appropriate manner. So again, the major histocompatibility complex will encode proteins that present this material to our immune system for recognition. And this is not an immunology course, but I just want to touch on some of these things because they're very important for our conversation. Now let's dig a little bit more into immune system development. Again, when we break down MHC, there's class 1 and class 2. Class 1 is also called A, B, and C. Class 2 are DR, DQs, and DPs. MHC class 1 is responsible for presenting material from intracellular pathogens to cytotoxic T cells, whereas class 2 presents antigens from extracellular pathogens to helper T cells to then orchestrate immune responses and promote antibody production or activate other immune processes. MHC class 2 is what is especially important for conversations surrounding bacteria presentation, right? So these are found on antigen presenting cells like dendritic, macrophages, and B cells. And as I mentioned, they're what's involved in engulfing extracellular pathogens. MHC is crucial in shaping self-tolerance and ensuring that T cells can recognize self-MHC molecules and prevent autoimmunity, as well as through negative selection, eliminating T cells that react strongly with self-antigens presented by self-MHC and contribute to self-tolerance. MHC is a set of genes found in all vertebrates, including humans, whereas the HLA is the specific term for MHC genes in humans. So it's important that you can distinguish between those two. And there are a wide range of MHC molecules. This is also important for things like organ transplantation. HLA does play a role in predisposition to various autoimmune diseases. There has been a link with DR and rheumatoid arthritis, as well as with juvenile idiopathic arthritis. You might find in the literature that we have a paper about this in a pediatric cohort. Type 1 diabetes is strongly linked to DQ and DR genes in class 2, as is celiac. And it's thought that in order for the likelihood of a person developing CD is significantly increased if they possess any of these high-risk HLA haplotypes. And there can be compounded risk if different alleles are present. And while HLA may play a role in these conditions, is not the sole determinant. Hence, a lot of interest in, such as in TEDdy, in finding environmental determinants that may play a role outside of genetics or compounded with genetics. And this is something that we saw in the JIA cohort, for instance, juvenile idiopathic arthritis. And this is also really relevant from the perspective of bacteria because MHC class 2 or HLA class 2 is what is very involved in presenting a correct immune response to certain bacteria. Thereby, it could play a role in bacterial colonization. And that's relevant not just in the context of something like probiotics, but just in how our body deals with commensal bacteria as well as pathogens. There are various immunomodulatory therapies that are based on modifying immune responses to manage disease. In some cases, this is very useful, but in other cases, it can present a lot of other complications for the patient, thereby increasing the need for better screening and also for a more diverse spectrum of therapies that maybe are a little bit less daunting and less difficult on a patient. Some of these immunosuppressants are really, really, really tough on the body. Genetics and the microbiome. So as we talked about the microbiome, there are a lot, and with the microbiome, there are a lot of players, a lot of determinants, a lot of factors that can influence composition. Genetics is one of them. In this case, genetics can shape the environment in which microbes thrive. Twin studies have shown similar microbes shared by genetically related individuals, But of course, in the social microbiome, we've found that social interactions, and this goes into lifestyle in the other slide, are incredibly important in influencing microbiome composition. And this is a concept that we'll go much deeper into in the animal microbiome module. But I did want you to recognize genetics plays a role, but also the social microbiome is super important. So who is residing in our household or near us, how we're interacting with each other, that plays a really critical role in what bacteria reside in and on our body. And host genes can, of course, also affect immune responses to microbes, such as in the case of HLA. And it's possible that genetic factors contribute to the development of disease by direct influence on the microbiome. So just reiterating that the factors influencing the microbiome are vast and also include genetics. This is a great article that recently came out, a large-scale association analysis looking at microbiome composition. And in this study of 24 different cohorts across Europe, Asia, and the Americas, the authors found that there were 31 different loci in a genome-wide association study. There were over 18,000 individuals across these 24 cohorts, and they found 31 loci or places in the genome that appear to have very, very strong associations with microbiome traits. This is a Manhattan plot for those who aren't familiar. It's like the skyline in Manhattan. So we're looking for those skyscrapers where a specific place in the genome along these chromosomes is popping through the threshold for significance, the p-value, and it's creating this skyscraper-like horizon. And so the 31 loci are here, and then the connection with a specific bacterial genus or family. So they refer to this as MBQTLs, microbiome trait loci. And what was really interesting about this is a lot of it made biological sense. So one locus that they found resides in the lactase gene, and this had study-wide significance, but showed especially an age-dependent association with bifidobacterium. This is intriguing to me because bifidobacterium is also increased in children who are breastfed longer, for instance. Other associations were suggestive, showing high heritability for genes that were highly expressed in the intestine and in the brain. So that's very interesting as well. Nutrition, environment, and metabolism domains were also highly implicated and suggested that there may be microbiome causal effects in ulcerative colitis and rheumatoid arthritis. This is a very interesting study that I'd like you to spend a little bit of time with this week as you have capacity. And what about you? What microbiome studies have you found that pique your interest? Please post them on our microbiome curiosity portal. Would love to hear more about what you're finding and what you're interested in, since it impacts all of us, right? So I would love for you to try to find your own articles and share with us either determinants of microbiome composition or examples where there was a strong association with a certain outcome or health measure. Would be very interested to see what you find and what you think is worth sharing. So in summary, the interactions are extensive and many different factors across a broad spectrum influence composition, and particularly in a compounded way. Genetics can play a role. And as I mentioned, the effect can be compounded, and this is explored in a variety of conditions. And here I've touched upon a few with respect to autoimmune disease, which is on the rise. And I do want you to recognize the role of HLA and that it is valuable in the conversation about what bacteria can colonize and how our immune system deals with bacteria that should or shouldn't be in our bodies. And this is important as well for self-tolerance and in the context of autoimmune disease. Future directions is another area where The microbiome curiosity could be very valuable. What approaches are you seeing in the literature targeting the microbiome based on genetics? Are there any? Or successful fecal microbiome transplants? What does the landscape look like for harnessing the microbiome to enhance health or to predict disease and thereby screen for it or treat it, prevent it? And also relevant are ethical considerations. So some of you have been noticing that there are certain underrepresented populations of people in these microbiome studies. And if ancestry is part of the major player behind microbiome composition, geographic location, all of these different factors play a role, then how generalizable are certain studies to other populations if we're not including them in our analysis? So there are a lot of ethical considerations to this, and I would love to hear uh, what you have to say and what you think about how we can improve how we study and how we think about these things. Thank you so much for watching. I also wanted to mention there's a lot of improvements in technology and how we sequence bacteria and viruses. So that's also relevant to the discussion as well. Thank you so much for watching. I look forward to your continued engagement in this course, and thank you very much. We'll see you soon.